ladies and gentlemen, and we appreciate your presence this afternoon for this important seminar in which our colleagues here is an intern from the Iowa State University. Sorry. And she stayed here for 2.5 months, yeah, 2.5 months, almost 2.5 months. And she did 2 excellent research that she will share to you. And hopefully that it will be a benchmark for other breeding activities. So without much ado, Ms. Carver.
Thank you, sir. So the first research we looked at was the response of germination to seed size and seed color of pigeon pea. So within pigeon pea, there are natural differences in seed size. The seed size is affected by variety. Thank you. Seed size is affected by variety, the environment that pigeon pea is grown in, and also different management practices. Farmers tend to select for larger seeds because they doubt the germination and stand establishment of smaller seeds. But if they were able to use smaller seeds, they would be able to reduce their input costs because they would be getting more seeds per unit weight. So we wanted to look at the differences between seed size and determine the effect of seed size on germination, and then also look at the effect of seed color on germination. We conducted Petri dish germination tests. We used 18 different genotypes. 2 were early duration and 16 were medium duration. We used 6 hybrids and 12 cultivars, and then we had the 2 different treatments of large and small seeds. We had 50 seeds per replication, so 50 seeds in each Petri dish, and then in the 18 genotypes there were 6 different seed colors that we looked at. The seeds were separated by 100 seed weight, so we hand separated the seeds and then recorded the 100 seed weight. During the germination tests, moisture was applied every 24 hours to ensure adequate moisture levels, and then germination percentage was recorded every 24 hours, starting at day 2 and going until day 8. So this is a table of the genotypes that we use, the 18 different genotypes. You can see the 6 hybrids, and then their seed colors and their 100 seed weight. Within each genotype, there was a significant difference between the small and the large seed weight within each genotype. So this is the analysis of variance for germination percentage, and you can see that genotype significantly affected germination percentage across all of the days. Seed size, however, affected germination percentage only up until day 7. So by day 8, there was no significant difference between the large and the small seeds. So first we'll look at the effect of seed size. Again, you can see from day 2 to day 7, the large seeds had significantly higher germination percentages, but by day 8 there was no significant difference, again, like I said, between the large and the small seeds. So when we looked at all 18 genotypes, there were 5 that stood out with the significant differences between the large and the small seeds. So ICP 11811, ICP 7035, ICEAP 000040-1, and ICP 13395 showed the large seeds had the higher germination percentages for the first 7 days. And then, like I said before, by day 8, there was no significant difference. With ICPL 8863, on day 2, the large seeds had higher germination percentages, but by day 6 and 7, the small seeds had significantly higher germination. And this was the only genotype where small seeds showed higher germination than large seeds. We also looked at the effect of seed color, so the 6 different seed colors and their effect on germination, and you can see that color only affected germination on day 2 and day 3. So by day 4, there was uniform performance between the colors. And this graph shows the germination between the seed colors. So you can see light brown and purple and black have the lowest germination percentages on day 2 and day 3. And then by day 4, there's no significant difference between the seed colors. So from this, we can conclude that there's a uniform performance between the large and small seeds with regards to germination by day 8. So this means that small seeds would be more advantageous for farmers to use because they can get more seeds per unit weight while maintaining the same germination percentage. And this would help them reduce their input costs as well. Also, there's a uniform performance between seed colors by day 4, which seeds are soaked in water prior to sowing. During hydropriming, seeds imbibe water, and this allows germination processes to start but not to be completed. It has been found to be an economical and simple and safe technique to increase germination, and also it is very applicable to resource-poor farmers because all that's needed is water. It has had mixed results in other species, and so we wanted to see if it was beneficial in pigeon pea, and if so, what duration of hydropriming would give the best results. So again, like I said, we wanted to determine the effect of hydropriming on the germination and seedling vigor of pigeon pea, and we also used 2 different soil types to determine the effect of soil type on seedling vigor. So we conducted a glasshouse experiment. We used 10 different genotypes, 2 were early duration, 8 were medium duration. We used 4 different treatments. So the control was not soaked at all. And then we soaked seeds in distilled water at room temperature for 2 hours, 4 hours, and 6 hours. So those were our other treatments. Again, we had 3 replications, and in each replication there were 5 samples. And like I said, we used 2 different soil types, the alpha soils and the vertisols. The experiment was watered daily, and we focused on 5 different observations. We looked at the number of seedlings that had germinated on day 3. We looked at how long it took to reach 60% germination. We looked at plumule length on day 8 and day 20, and radical length on day 20, which would be indicative of seedling vigor. So again, these are the genotypes that we used and their characteristics, 100 seed weight, and their seed color. And this is the analysis of variance for all of the observations we looked at. Soil type, genotype, and the interaction of soil type and genotype were significantly affected each characteristic. Hydropriming had a significant effect on plumule length in day 8, radical length in day 20, germination on day 3, and then days to 60% germination. So that's what we'll look at first, is the effect of hydropriming. You can see here that the 4-hour treatment had the highest number of seedlings germinated on day 3. And in the same way, the 4-hour treatment took the least amount of time to reach 60% germination. For the plumule length on day 8, again, the 4-hour treatment showed the best performance, having the longest plumules, and then 6 in the 6-hour treatment in the control performed significantly worse. With the radical length, the control, the 2-hour treatment, and the 4-hour treatment were all actually, there's no significant difference between them. But the 6-hour treatment showed significantly shorter radicals than the control and the 4-hour treatment. But looking at all 5 observations, the 4-hour treatment performed the best with all 5. So next we looked at the effect of soil type. So with regards to seedlings germinated on day 3, the alpha sol had more seedlings germinated than the vertisol soil. And in the same way with the days to 60% germination, the alpha sol took less time to reach 60% germination than seedlings in the vertisol soil. With day 20, seedlings in the alpha soil had longer plumule and radicals. However, on day 8, the seedlings in the vertisol soil had longer plumules. But again, looking at all 5 observations, seedlings in the alpha soil performed the best. Then we looked at all of the 10 different genotypes that we tested. ICP 14304 and ICPL 87091 had the most number of seedlings germinated on day 3. And those same 2 genotypes took the least number of days to reach 60% germination. With regards to seedling performance, ICP 11811 and ICP 7035 showed the longest plumules and radicals. And then looking at the interaction between hydropriming and genotype. So you can see across the genotype, again, the 4-hour treatment had the most number of seedlings, usually had the most number of seedlings germinated by day 3. With regards to days to 60% germination, again, the 4-hour treatment performed the best across the genotypes, showing that it took less time to reach 60% germination. And you can see that the control usually took longer to reach 60%. The interaction between soil type and genotype. So again, like I said before, the alpha sols usually performed better with regards to seedlings germinated on day 3. And then same with days to 60% germination, the alpha sols took less time to reach 60% germination across all of the genotypes. So with regards to plumule and radical length, for day 20, again, the alpha sols showed the better performance with the longer plumules and radicals as compared to the vertisol soil. But again, with plumule length in day 8, the vertisol actually showed longer plumules across the genotype. And then the interaction between all 3 factors, so soil type, genotype, and hydropriming, had a significant effect on plumule length in day 8. So this is the interaction between all 3 factors. So you can kind of get the idea that the 4-hour treatment is usually showing longer plumules in day 8 than the other treatments in the control. So from this we can conclude that hydropriming allows for faster seedling emergence. Pigeonpea is starting to be planted after a rice crop in rice fallow systems, and so if hydropriming allows for faster seedling emergence, it can start utilizing that available moisture quicker than if it would take longer to emerge. Within hydropriming, the 4-hour treatment showed the best performance, like I said, showed longer plumules and longer radicals, which could lead to more drought-tolerant seedlings. It also showed faster emergence, which would again lead to a successful stand establishment. Pigeonpea grew better in alpha sol soils as compared to vertisol soils. ICP 11811, ICP 7035, and ICP 87091, they performed the best with regards to plumule length and radical length. And then a recommendation for the future would be to conduct this experiment in a rice fallow system to see if hydropriming was beneficial to that system. So before I open the floor for questions, there's just a few people that I have to acknowledge. The Plant Sciences Institute at Iowa State University, they funded my time while I was here, so I'm very thankful to them for allowing me this opportunity. The Learning Systems Unit here at ICRISAT, I'm thankful that they just welcomed me here and got me set up with this internship. The Biometrics Department and Dr. Ratour analyzed all of our data, so I'm very thankful to him. And of course the Pigeonpea Department, so thankful for the encouragement you gave me, especially to Polly for all of her help with both of my experiments, and Dr. Mullah for allowing me to work under him and learn from him, and just grow a lot during my time here. So I'm very thankful to all of you. And with that, I'll open the floor to any questions that anyone may have.
Yeah. Okay, it's now open for questions and answers. So, by the way, I will add more in the introduction. Kathy is a undergrad, third year college undergrad, taking up the major in agronomy.
Did you study relationship between seed size and radical plumage growth?
No, no. We just looked at germination and
Because,
seed size.
Because for example, 7035 is large seed, big cotyledon.
Mm-hmm.
So the seedling have that advantage, initial advantage,
Mm-hmm.
which dissipate after some time. So that kind of things can happen. Now, another point is that you mentioned 4-hour priming is good, especially referring to rice fallows.
Mm-hmm.
So maybe Maya can also answer what kind of moisture we have in rice fallows at that time, so that it can pull off not only germination but its seedling have to pull out. So that kind of moisture is enough in rice fallows. If you plant seed with priming, 4-hour priming, will it be beneficial? Seedling will come quickly or not?
Yeah, that is the idea here, is to find out, is to find out if the availability of the moisture in after rice is enough for the pigeon pea seeds to germinate. In our experience, what we have observed is if we sow the normal sowing, okay, and it was observed that it's taking us 12 to 15 days to germinate the seeds. So meaning if you calculate the 15 days that the seeds to germinate, then the evaporation of the moisture in the seed is evaporating, not used by the plant. Yeah. But with this idea, in case we can do it in the rice fallow, then we can see that hydropriming can quickly germinate the seeds quickly, utilizing the moisture in. Then after that, we can utilize directly the moisture that is left in the field. So this is the idea that we are. Well, literature says that the moisture is capable of supplying the next crop for 2 months, something. But we have not yet done this kind of experiment. But we hope that we can do this and check really the moisture content, whether it's a black or red soil.
Time to ask questions, okay?
Yeah, sure.
No question on this, because you are the expert. How was your stay here with in terms of learning more about pigeon pea, other than what you have reported?
Yeah. To be honest, I'd never even heard of pigeon pea before coming to India. In Iowa, where I'm from, we grow maize and soybeans. So pigeon pea was a totally new crop to understand and learn about. So definitely just those first few days of what even is this plant? What is it used for? How is it grown? How long it takes? Learning all of that was very beneficial. I now have the book that about pigeon pea to take back to the States and educate people back home about pigeon pea and its use for resource poor farmers. So yeah, it was very good. Yeah. Yes.
Mm-hmm. From who's the leader from here? Okay, I'll read. I quote. Why do you think that color of the seed can be associated with the germination?
One of the literatures I read said that black pigmented, it was in soybean, black pigmented soybean imbibed water slower than non-pigmented soybean. So with the purple and the black pigeon pea, that would make sense where it imbibes water slower, so it takes longer to germinate. Light brown, though, showed the lowest germination percentage, or took the longer to germinate, so I'm not sure why that would be. It could be the genotype or something like that.
Yeah, that will be another in-depth research.
Mm-hmm.
At least there is a, we have started this, so let other scientists also do this. Any more?
Okay.
Please enjoy this session.
You like to work for masters on this crop,
For what?
for a master's degree on this
Oh,
crop?
it would be interesting. There's a lot of potential, I think, for pigeon pea. So, yeah, yeah. Huh?
Okay, as a backgrounder of this research, with the help of Ravi Kumar, and with our search on previous researches, there's nothing coming out of this type of topic. So another topic is coming out. So I think this will also help other in the breeding to look into this type and then do some other type of research. Do you understand?
Yeah.
You have shown the effect of hydropriming on radical length and this will the right?
Uh-huh.
Hydropriming effect on plumule length and radical length. That you show that there is effect on plumule length but not on any radical length.
Mm-hmm.
How do you explain that? Because when being pigeon pea a drought tolerant crop, it's expected its radical length should go deeper.
Mm-hmm.
I think
This
effect
one?
of hydropriming on plumule length and radical length.
There's plumule length. There's radical length. Are these the two that you were asking about?
Effect of hydropriming.
Right. And then radical length, right?
So there was effect on radical length.
Actually, there's no difference.
No difference.
soaking,
Right.
4 hours soaking
Right.
is same.
Right. And same as 2 hours as well.
And also the plumule length, you see there's no any defect.
What would you think? What happened after 6 hours? It's very rotting or something, right?
In other literatures that have said that over-priming, so priming for too long, there's been oxygen deficiencies and buildup of inhibitors, so that could possibly be a factor.
Okay, with regards to your question, yeah. we think that because this is take note that this is a 20-day, okay, so very short period. It's a short period that we can measure the radical length as per the plumule length. So maybe if we can have a longer period, like we can get 2 months, then maybe we can have a complete scenario of the relationship
It's
between
too
these
early
two.
to make any.
Yeah, because it's too early, because it's too short also for her to stay here. But at least there's something story behind.
Mm-hmm.
I think you have done a very good job in such a short time.
Yes, thank you.
And very good work. I think we were discussing earlier that from here he is going to take lead to really develop a technology for rice fallows, which is very critical, and millions of hectares are available for that, not only in India but other countries as well, where moisture is really limiting factor.
Mm-hmm.
So I think you have made a good beginning. Let us follow it up, and we will offer you postgraduate. So you think when you're ready for that, Meyer still will be here.
Yeah. Thank you, sir.
Yeah, so I think we start a new beginning in our program. Yeah. Really, thank you very much for coming to this place, choosing this place, and spending time with us.
Yes, thank you for allowing me to. It was a really great experience.
Okay, no more questions. We thank you very much, Katie, and good luck for your professional growth. We hope to see you.
Thank you. Thank you.
Before we end, we'll just put some photo session of the certificate.
All right.